Okay, we're 2 minutes past the hour, so I think we will start because I've got a short introduction anyway, and as more people are joining the webinar, I'll be going through that in preparation for the opening keynote talk. So for all those who've joined us, you're very welcome. My name is John Morrissey. I'm from University College Cork in Ireland, and I'm the moderator for today's session. Now I'm going to share my screen and give you I have a short little introduction here. So today is a second webinar in a series of 3 on microbial cell factories that's been organized by 2 European Union funded projects, TOPCAPI and CHASSY. I'm the coordinator of the CHASSY project, and Professor Enrico Taccano is the coordinator of TOPCAPI. And both of these projects are systems biology stroke cell factory, microbial cell factory projects. So we decided that we'd like to organize this webinar series on the theme of biological principles to commercial products. Initially, we had actually planned on organizing a conference, but obviously that was no longer possible. So there are 3 webinars. In the previous webinar that Enrico moderated, she gave a brief introduction to TOPCAPI. So I'd like to do the same for CHASSY. If I just minimize some material here so I can see myself. So the CHASSY project is a 4-year project funded by the European Union, and the aim is to develop efficient yeast cell factories for industrial production of novel high-value products. And what you can see on the screen here are the various partners who are involved in CHASSY. There are some industry partners, Clariant Biopetroleum, and a range of academic partners. And I've just listed the lead scientists and lead PIs at each of the institutions here. In all the cases, there's actually quite substantial teams involved in the research that's ongoing in the project. I'm not going to say too much about the project other than, I suppose, the concept of the project is very straightforward. We're working with 3 different yeast species that you see on the left: Saccharomyces cerevisiae, Eurovia lipolytica, and Cluromyces marxianus. And we've been developing systems biology and synthetic biology approaches to optimize these strains for the production of oleochemicals and aromatics. And the reason for this is that these are molecules that can have a lot of applications in the bio-based economy and indeed in the biopharmaceutical sector. For those of you who attended the first webinar, you'll know that Ivan Domenzin from Chalmers University presented a lot of the work that we've done on modeling, and he would have presented the EC container, where there's a whole collection of new enzyme-constrained models of organisms, including the 3 that are listed here. So if you didn't see that webinar, you can get it on the same website that you registered here. But focusing in now on this webinar series, what we're really about in these projects and in this series is the idea that we can have sustainable production of a whole range of different bio-based molecules that are useful in a range of sectors, and for products in a range of sectors, like you see illustrated here on the right, ranging from foods and nutraceuticals to bio-based chemicals, pharmaceuticals, and cosmetics and flavors and fragrances. And this concept is founded or centered on the biorefinery concept, where one would use microbes to take in feedstocks, ideally sustainable feedstocks, and make a whole range of different products. But for this, we need very effective biorefineries, and we need very efficient cell factories. And the cell factory organisms, they need to go through this kind of this sequence from the design of the actual cell factory, the construction of the cell factory, and ultimately the validation and scale up. And we decided to organize these 3 webinars around that theme. So on the 11th of March, we had a webinar based on the design concept. which you can see, as I said, on the registration website if you missed it. Today we're focused on the build component, and I'll introduce our speakers in a moment. And on the 8th of April, we've got the validate stroke scale up component that will be discussed by experts in that area. So in each of these webinars, we've got 3 speakers, an invited keynote speaker who is a world-renowned expert in each case in the area, and then 2 equally important speakers, one from each of the host projects, CHASSY and TOPCAPI, and those speakers present some of the key outcomes from either CHASSY or TOPCAPI on the topic of the particular webinar. So I'm really honored to be able to introduce today's keynote speaker, who is Professor Huimin Zhao. Following his graduation in China, he carried out his PhD in Caltech before taking up his position in the University of Illinois, where he's a Stephen Miller Chair of Chemical and Biomolecular Engineering. Professor Zhao has an enviable publication record in the area of metabolic engineering in a whole range of different organisms, which is something that's maybe a little bit unusual in the field, where many people focus on 1 or 2 organisms. In Professor Zhao's case, if one looks at his publication record, many different organisms, and also a whole range of different technologies. You know, there's rational strain engineering, but also systems biology and genome scale modeling, multiplex approaches and high throughput ways of engineering strains. I think he's going to give us a fascinating insight today on his, hopefully, some of the most exciting developments that are going on in his team. I'll introduce the other 2 speakers just before their talks. In terms of a little bit of organization and housekeeping, the speakers will have 20 minutes or 15 minutes for their talks. After that, after each talk, we have time for about 5 minutes of questions. So if you have questions, please type them into the Q&A box. You can type them in as we go through or anytime, but we will just answer them in the Q&A section. The chat function is disabled, and so speakers won't be answering questions in real time. Either myself or Enrico will read out the questions to the speakers at the appropriate time. Then after the 3 presentations, hopefully people will think of more questions that they would like answered, or maybe questions that they didn't, that there wasn't time to answer in the initial Q&A. And we can pick those up in a general discussion with all 3 panelists. So I think that's my introduction covered. So I'm going to stop sharing my screen now, and I will hand the floor over to Professor Zhao. Thank you.
Okay, thank you for your nice introduction, John. Today, actually, I'm going to talk about recent work in the engineering of microbial cell factories for production of chemicals and materials. My research program at the University of Illinois is focused on synthetic biology. We're very interested in developing foundational tools for protein engineering, metabolic engineering, or genome engineering, etc. And then we try to leverage those tools for 3 main applications. One is related to the discovery of novel natural products as bioactive compounds or drugs. And also, we are interested in engineering microbial cell factories for production of chemicals and biofuels. And today, of course, many talk about that topic. And then lastly, we're also interested in the application of those synthetic biology tools for mammalian cells as well, particularly for gene therapy and also fundamental studies like the chromatin structure and dynamics. So a few years ago, we built a Biofoundry called the Illinois Biological Foundry for Advanced Biomanufacturing, or iBioFab in short. So our idea is to basically integrate many instruments together and to address the various applications that we face, in biotechnology, such as protein engineering, pathway engineering, genome engineering. But as we know, those workflows are very complex. So we tried to break those workflows into process modules, which will be in turn broken down to unit operations. So then we can program those unit operations to come up, custom designed workflows for different projects. And so we basically integrate, more than 20 instruments on this large platform. In the center of the platform, there's a robotic arm that will take place from one instrument to another. So essentially, we kind of developed an automated laboratory for bioengineering. So we try to leverage this platform and in combination with the synthetic biology tools for metabolic engineering. So the first thing we did actually is mainly for protein engineering. At that time, the CRISPR technology was not developed yet. So we developed a fully automated workflow for talent synthesis, which is actually a powerful genome editing tool at that time. And as you know, talent actually consists of the DNA binding domain and a nucleus domain. And the DNA binding domain consists of many repeats. Each repeats will recognize a particular sequence. So what we did is to prepare a library of those repeats, more than 300. And then based on the target sequence, we can choose those parts and assemble them to create an artificial DNA binding domain, which will be linked to the nucleus domain to create those talents. And we developed this workflow that can be implemented in like 24 hours using the robotic system that include the assembly of the parts, the DNA transformation, and then also the plasmid purification. So as a result, we can significantly reduce the cost for talent synthesis. For us, it only costs like $3 for one talent. And of course, nowadays, most people use CRISPR, but I would say the talent still has some niche applications. One application, for example, is shown here, is for the treatment of mitochondrial disease using talents. Now, it's very hard to deliver the CRISPR into the mitochondria, but the talent could be readily delivered to the mitochondria because we just need to use a mitochondrial localization sequence. And in this work, collaborative work, we showed that the engineered talents could be used to treat mitochondrial disease very well. And also more recently, we showed that actually the talents can work very well, work better than the CRISPR in the heterochromatin region. Right? Most people, of course, use the CRISPR for genome editing, and those genes probably are in the euchromatin region. But we actually use a single molecule tool to study the search mechanisms of the talent, and also compared with the Cas9. And we found that they do use the different search mechanisms. And we found that the talent will search the targeted sequence much more efficiently in the heterochromatin region. So we predicted that it will work better in the heterochromatin. And indeed, we proved that it works better on those targeted genes in the heterochromatin region. And of course, we're not just interested in building like talents, right? We're mainly actually try to use the synthetic biology tools and the Biofoundry for metabolic engineering applications. So a few years ago, we collaborated with ADM to help them to optimize a biosynthetic pathway involved in the synthesis of an amino acid. So in this case, we used a similar kind of the DNA assembly tool used for talent synthesis to build a library of pathways that are involved in the synthesis of a particular amino acid. And we constructed more than 1,000 pathways just in a matter of a few days. And this library consists homologs from different species and also promoters with different strengths. So essentially, we create a combinatorial library. And that, of course, is managed on the library construction part. But then a few years ago, we thought that whether it is possible to really develop a fully closed design, build, test, learn cycle And in order to do that, of course, we also have to incorporate machine learning into it, because otherwise, the researcher have to decide, right? And that will not be able to fully automate the entire design, build, test, learn cycle. So as a proof of concept, we studied a Lycopene pathway that consists of only 3 genes. And we used a robotic system to construct, hundreds or thousands of those pathway variants. And then we analyzed the results. The Biofoundry actually will purify the product and then analyze the results, right? And then we used the machine learning model to predict what kind of combinations of the promoters could lead into higher production of lycopene. And then the Biofoundry will decide, what the library it should construct in the next round. So then the whole, design, build, test cycle could be fully automated. So I just mentioned the Lycopene pathway is relatively simple. It only consists of 3 genes, right? And we use the library of promoters with the different strengths. And in this case, we picked the 24 promoters for each structure gene. So then, as you can see, the total combination for these promoters will be about 13,000. And our goal is to basically search the library to find the best combination. And in order to do it in an automated manner, we actually developed a fully automated workflow for the entire pathway engineering process that includes the construction of the pathway, the transformation of the pathway library to the E. coli, and followed by the extraction of the product, and then and also the detection of the product, and then also the analysis of the product profiles. So basically, it's all the steps involved in the pathway construction analysis. So in the first round, we constructed we picked the 46 library, 46 variants, and then analyzed the production of the lycopene. And then based on that data, we predicted what kind of the promoters could be used to improve the production of lycopene. And then we actually constructed a new library again, and then go through the whole process again. So we repeated this cycle for like 3 times. So in total, we screened about 150 clones, right? And so in the first round, we basically kind of randomly searched the sequence space. But in the second round, we actually construct kind of a biased library. And in the third round, we can further construct a biased library based on the second round of results. So in this iterative process, we can gradually improve the production of the lycopene. But the key point here is this essentially still is a kind of a direct evolution approach. But I think the key point here is that we can screen a very small library, right? Instead of screen the more than 10,000 variants, we can only screen less than 200 variants. Right? In each round, we only screen the 46 variants, and yet we can find the optimized pathway variants very quickly. Actually, we did also do a control experiment. We just randomly pick those variants and do the screening, and we can actually find the one that better than what can be found by that random approach. And of course, this is for pathway engineering, but our ultimate goal is really for the metabolic engineering, right? It's not just the pathway itself. Also, we will try to identify the targets on the chromosome that can affect the production of the compound. So in this case, we had the vision that we try to develop a automated metabolic engineering platform. We call it automated cellular engineering platform. So the idea is to use those genome scale engineering tools like CRISPR or RNA interference, those tools to perturb the gene expression on a genome scale, and then try to screen the library to find the improved variants, either using high throughput screening method or in vivo biosensors, and then try to incorporate those beneficial mutations into the chromosome, and then repeat this cycle again and again, right? So essentially, it's this direct evolution concept. And in metabolic engineering, there are kind of two traditional approaches. One is the adaptive evolution that actually works very well, but it only limited to the phenotype that can be linked to the cell growth. And another approach is the rational design. Most people use that approach. And basically, we try to use system biology tools to identify those rate limiting steps, and then use the genetic engineering tools to make the desired mutations. And this is an iterative process as well, but it's very time consuming. So about 10 years ago, we started thinking about whether it's possible to do it in a much more efficient manner, particularly is try to identify those non-intuitive mutations in the cell that can affect the protein, affect the product production. And initially, we thought about the Rna interference method because there's no other method available to perturb gene expression on a genome scale at that time. And then later, when the CRISPR technology was developed, then we tried to use that method for genome scale engineering. And so initially, we thought about the RNA interference approach, and it is actually pretty simple. Essentially, we just expressed the RNA interference machinery that consists of only 2 genes in Saccharomyces cerevisiae, and then we can isolate the genomic DNA and create a library and to prepare the RNA interference cassettes for each targeted gene. There are 6,000 genes in the baker's yeast, and then we can screen the library for the improved phenotype. But RNA interference is only for gene downregulation, right? But we also wanted to overexpress those targeted genes. So then we came up an idea to use the cDNA rather than the genomic DNA to prepare the RNA interference library. So we can overexpress those cDNAs to create the overexpression library for most of the genes in the host, and also use the RNA interference library to do the genome-wide knockdown. So then we can transform these 2 libraries to yeast to find the improved phenotypes. And we also wanted to leverage the Biofoundry we have. We developed a fully automated workflow for metabolic engineering. So that actually took us also a long time to develop. That is more complicated than the pathway engineering workflow. That include the construction of the RNA interference or overexpression library, and followed by transformation of the yeast and also the high throughput screening. But using this platform, we were able to identify some variant, the mutant strands that showed significantly improved tolerance to an inhibitor that commonly found in the lignin cellulosic hydroxide, which is the acetic acid. And through 3 rounds of evolution, we were able to identify one mutant strand that shows the highest reported acetic acid tolerance. It is more than 1% tolerance to the acetic acid. The whole workflow takes about 2 weeks to implement. And as I mentioned, this is essentially the direct evolution approach for metabolic engineering. And the direct evolution field actually was studied in the early 1990s, right? Initially, the direct evolution approach was applied to protein engineering, and then later applied for pathway engineering. And only more recently, it was applied to metabolic engineering. And one landmark work was from George Church's group, which is the development of the MAGE method for E. coli engineering. And also in the recent years, more groups actually try to develop those in vivo continuous evolution approaches, and also genome scale engineering approaches for metabolic engineering. And I was very fortunate to join to be part of this field very early on when I studied my graduate school. So my former advisor, actually Francis Arnold, got a Nobel Prize in 2018 for the pioneering work in direct evolution. And currently, I'm also actually involved in a DOE Bioenergy Research Center that is focused on the engineering of microbial cell factory, which is very similar to the research program in CHASSY. So we are also focused on yeast, particularly non-model yeast, and also the model yeast, the Saccharomyces cerevisiae. So we try to build new capabilities for the Biofoundry, particularly focused on the design, build, test, learn cycle, right? Those. But one notable difference is that really we also try to develop machine learning algorithms for protein engineering and metabolic engineering. And also we develop many genetic tools for those non-model yeasts. And we're particularly focused on like, the CHASSY, the lipid-based chemicals, and also some organic acids in the Isachenkia orientalis. And then also just a few months ago, I got a NSF Center grant that is focused on the development of machine learning AI tools for the molecular synthesis. And we are trying both in chemical approaches and the biological approaches for the production of chemicals and materials. And I think this is also emerging area. So we try to leverage the Biofoundry we have. And in addition, we have the automated synthesizer for chemical synthesis as well. So we have a very strong team, but most of us also don't have all the the expertise yet. So we are learning as well. So in summary, what I show today is that we actually established a Biofoundry for next generation synthetic biology applications, and I showed a few examples. We can use it for the construction of talents at a very low cost and efficient manner. And also I showed that we can use the Biofoundry for pathway engineering. Particularly, I showed that we can incorporate machine learning to actually develop a fully closed design, build, test, learn cycle for pathway engineering. And lastly, I showed that we develop many genome scale engineering tools for metabolic engineering. But I think if we can also incorporate the machine learning, then we could also develop a fully closed design, build, test, learn cycle for metabolic engineering as well. And I strongly believe that there are many opportunities actually at the interface of synthetic biology, machine learning, and automation. There will be more advances in the next few years. So finally, I would like to thank the students who did the most of work. Because of time limitation, I will not mention their names, and also thank the collaborators. Finally, I would like to thank you for your attention, and I'll be happy to answer questions.
Thank you very much, Huimin. That was hugely impressive, and it looks like you're keeping yourself quite busy. So we have a couple of questions coming in. So the first one here relates to the fear of all the scientists. Do you see in the future that the wet lab scientists will be completely replaced by these biofoundries?
No, I think it's not. Actually, it will be the opposite. So actually, we will find more opportunities to do. And then also the scientists will spend more time on the design and the learning part. Right? And even with the development of machine learning, I don't think the machine can help us design everything, right? And I think we still need actually develop new knowledge for the design and the learning part.
And a question for me that's maybe as a follow-up. I mean, after you go through, let's say, the automated strain construction and with the machine learning and everything else, and you end up with your best strain. I mean, is that typically a final strain, or would you say that's more a lead strain for maybe further specific development?
That's right. That's right.
So I mean, and that's a case where presumably then you will need a wet scientist and the brain power as well coming in, getting involved.
Yeah, and basically we have to also do more characterization as well, right?
Yeah.
So this automated platform is mainly just for the construction of the strain, but then later I also have to do more characterization to learn the engineering principles or the mechanisms. Right?
Yeah.
And also, I remember when I was a graduate student and I had to pull the gel to run the DNA sequencing by myself, right? And nowadays, we can just send it out to the vendors to the all sequencing, but yet we find more things to do, right? So that's why I don't worry about that at all. And I would say there were more things for the experimentalists.
Absolutely, and we can do more useful things. Even though I spent many a happy afternoon reading gels, G, A, G, G, T. I mean, it's quite therapeutic, but no, I think we can do more useful things. So great. There's a question here. Which aspect is more important, pathway optimization or chassis optimization? And where's the bottleneck?
Yeah, I think probably the chassis optimization is more important than the pathway optimization. Because for pathway optimization, it's pretty straightforward. Many you try to just find the best enzymes, right, that works well inside of this host, and also the promoters, right. There are also there are many promoters available. So I think you can optimize the pathway using the workflow I just mentioned, basically for almost any pathway. But for host engineering, that's very difficult, mainly because it's a highly interconnected network. It's very hard to actually identify those non-intuitive gene targets. For the obvious one, we all know we can definitely try right away, and those are obvious. Just look at the network. But if you really want to find those non-intuitive, many of those synergistic kind of interactions that are not directly in the pathways linked to the product, then it's very hard to predict, right?
Yeah.
So that's why I think it's much more challenging, especially if you want to commercialize the process. Because in publication, we just need to show some improvement, and that's still very easy. But if you really want, let's say, reach commercialization, you have to, reach a target titer, and it will be very difficult. Even you want to improve like 20%, it will be very, very challenging.
Yes. I suppose a related question I have on that is, I mean, do you think the chassis optimization and the pathway optimization are completely separable?
Yeah, I think they can be separated to a certain degree, but in the end, I think we still need to optimize the pathway in the context of the host, mainly because the enzymes in the pathway will also interact with the genes in the host, right? They're not separate.
Yeah. Okay, and I'm just going to take two more questions, and then we'll have to save other questions for later. Yeah, this is maybe a follow-up question to your point just now, but the question is, how much does chassis non-optimality limit a choice of hosts to a few model species? I think it's really talking about this use of maybe non-model organisms and new non-conventional species. And will synthetic biology overcome this limitation?
Yeah, I think this is a very good question. And recently, we wrote a recent review on this topic. And I believe that with the recent development of the CRISPR technology and also other kind of related technology, I would say more and more researchers will start to work with the non-model yeasts. Because in the past, I think the one major limitation is with non-model organism is the lack of genetic tools. But now I think we have kind of the blueprint to create those tools very quickly. Usually, typically, probably less than a year, right, for any given target organism. So I'm very optimistic that more and more non-model organisms will be explored in the future, and they actually give us a head start in metabolic engineering. Because we know that all those non-model organisms have unique physiology and also have some unique kind of product profiles.
Okay, that's great. I'm going to have to stop this particular discussion, but one of the things just to flag for you, Huimin, that I'd like you to think about, I'm sure you have before, but for the final discussion as well, is how we kind of move this technology from, let's say, the research phase into industry, especially with non-model organisms. And so I'll ask you about that in the general discussion afterwards, because I think we're going to have to move on to the next speaker now. So thank you very much. And
thanks. Yeah.
So I think you're able to stop sharing your screen now, Huimin. And our next speaker is going to be Dr. Mislav Oreb. Mislav is a group leader at Goethe University in Frankfurt. Mislav's main research in recent times has been with Saccharomyces cerevisiae, so the traditional yeast, if I call it that. But Mislav has been doing a lot of non-traditional things with this particular organism, and is interested in metabolic engineering, development of biosensors, the application of synthetic biology. He's done some really nice work on transporters over the years. But I think today he's going to focus in on some of the work that he's been doing on the lipid side of things, and on the synthesis of fatty acids and the engineering of yeast for that purpose. So Mislav, thank you very much for agreeing to speak today, and the floor is yours.
Thank you very much for the nice introduction. So I'm sorry. Still not in my first slide. Sorry, I will stop the sharing.
That's fine. We'll save a lot of time if you just go straight to your conclusions.
Yes.
I see that my co-organizer, Professor Rico Tacano, has joined the meeting as well. So Rico will help me out with the questioning too, I think.
So I hope you can see my first slide now.
Perfect.
Great, thanks. So fatty acids, which I will talk about today, are very versatile platform chemicals having hydrocarbon chains of variable lengths, which is indicated here by an R, and the terminal carboxyl group. They can be converted chemically or enzymatically to different related compounds, such as fatty alcohols or esters, decarboxylic acids, or alkanes, for example, to mention just a few, which have wide applications in industry ranging from biofuels to pharmaceuticals. Traditionally, these products are manufactured in a petrochemical industry, and also by derivatization of fatty acids, which are extracted from plants, for example, from oil palms. And nowadays, there's of course an increasing interest in more sustainable and also potentially more efficient methods to produce fatty acids and derivatives thereof. So in the CHASSY project, we are also focused on two fatty acid-based products. The one is octanoic acid, which is a medium chain fatty acid consisting of 8 C atoms, which has potential applications as an antimicrobial agent, but also as an additive to feed or pharmaceuticals, and also as a platform chemicals, which can be used to derive, for example, C8 decarboxylic acid, which in turn is a precursor of biopolymers, or N-octanol and heptane, which are potential drop-in biofuels. Also, it can be derivatized to esters, some of which can also be used as additives to perfumes, for example. And the other product is docosahexaenol, which is a very long chain product. It is a very long chain alcohol, which has antiviral properties, for example, against the herpes virus, but it is also used as emollient or emulsifier, or as an additive to cosmetics and nutritional products. So both products have potential industrial applications and are already used in the industry. So in Goethe University, we focus on the octanoic acid. I will talk only about this part today because the time is rather short. So I will present this as a case study for production of fatty acid-based products. So we are engineering Saccharomyces cerevisiae, and this first slide shows very simplistically how fatty acids are produced in yeast. So we have a fatty acid synthase type 1 in yeast, which is a macromolecular complex with many functional domains. And this complex uses simple precursor molecules, acetyl-CoA and malonyl-CoA, to synthesize the hydrocarbon chains of fatty acids. NADPH is used as a reducing agent. So this cycle starts with loading acetyl-CoA as a SATA unit, and subsequently malonyl-CoA is added as an extender unit to extend the carbon chain by 2 carbons per cycle. So this is repeated in an iterative manner. And in Saccharomyces cerevisiae, usually the products which are released are fatty acyl coenzyme A esters in a chain length range from C16 and C18. So now our idea was to reprogram this fatty acid synthase to produce octanoic acid and also other medium chain fatty acids. And the group of Martin Gröninger here in Frankfurt, they're working on the structure of fatty acid synthesis, and based on the structures, they suggested mutations in specific domains of the fatty acid synthase, which could introduce the desired properties, which is the synthesis of shorter chain lengths. So we specifically targeted the acetyl transferase domain, the keto-acyl synthase domain, and the malonyl palmitoyl transferase domain. Not the other domains are not shown here, which we did not mutate. So when we looked into the product profile of this mutated fatty acid synthase, we could see that most of them produced medium chain fatty acids at various amounts and various ratios. And some of them were quite specific, either for hexanoic acid, C6, or for octanoic acid. And this is the one which we are interested in. So it produces also minor amounts of hexanoic acid, but the main product is octanoic acid. So we selected this mutant to engineer our chassis strains. So the mutation is located in the malonyl palmitoyl transferase domain, and this domain has a dual function. On one hand, it loads the extender unit, malonyl-CoA, and on the other, it releases the product, which is the acyl coenzyme A ester, in our case, octanoic-CoA. And the rationale behind this mutation was to decrease the affinity for the malonyl moiety. So we introduced lysine instead of an arginine, which means a shorter side chain. And in this manner, the interaction between the amino acid and the malonyl moiety is weakened, which in turn leads to disfavoring the extension of the fatty acyl chain. So this should result in the production of shorter chains. This was the idea behind, and also an explanation how this works. Okay, now we went on with the traditional metabolic engineering, which was discussed also before. So we engineered first the logical targets. Then we introduced our mutated fatty acid synthase into yeast. It releases octanoic-CoA esters, and these esters become cleaved by thioesterizes. There are multiple thioesterizes in yeast which are able to do this, and they are quite promiscuous in their substrate range, so that the octanoic acid can be secreted as a free acid out of the cell, which is of course beneficial for purifying the product. Along with this mutated fatty acid synthase, we could achieve some production of octanoic acid in culture supernatant. So you can see it's about 30 milligrams per liter without any further engineering of the chassis. So next we went through the logical metabolic engineering targets. One idea is, of course, to prevent the degradation of the free fatty acid, which in yeast occurs in peroxisome. So not only octanoic acid, but also others are degraded through beta oxidation, which is located in peroxisome. And free octanoic acid, which is transported inside the peroxisome, first becomes activated by this fatty acyl synthase, CoA synthase, Fa2, to octanoyl-CoA, which then becomes further degraded in the beta oxidation cycle, and the process is initiated by the BOX1 gene. So the idea was to delete these two genes to prevent the degradation, and this indeed led in a cumulative manner to an improvement of the production by approximately a factor of 2 already. So next we looked at the precursor supply. I already mentioned that acetyl-CoA and malonyl-CoA are necessary to synthesize fatty acid, and also the NADPH as a redox cofactor is necessary to reduce the hydrocarbon chains. So engineering precursor supply and cofactor supply is usually overlapping tasks because both acetyl-CoA and NADPH are delivered by the central carbon metabolism. In Saccharomyces cerevisiae, acetyl-CoA is synthesized through the so-called PDH bypass. PDH stands for pyruvate dehydrogenase. So this pyruvate dehydrogenase bypass, which takes place in a cytosol. And this pathway converts acetaldehyde, which is derived from the glycolysis, first to acetate. So it's first an oxidation step. And next, this acetate is ligated with coenzyme A in an energy-dependent manner. So we need ATP in this reaction, which is not desirable from a point of view of a metabolic engineer, as energy metabolism or energy yielding metabolism competes with the production pathway for the carbon source. So we reasoned that introducing a heterologous pathway, which is energy independent, would improve the production of octanoic acid. And we selected for this the phosphoketolase, a variant which is specific for xylulose 5-phosphate. And this enzyme converts xylulose 5-phosphate to glyceraldehyde 3-phosphate and acetyl phosphate. And this acetyl phosphate is further converted to acetyl-CoA by acetylase. So we introduced these two genes into our strain and thereby disrupted also the GPP1 locus, which encodes an enzyme that converts acetyl phosphate into acetate. And this is, of course, a not desired reaction in this place. So we delete this gene and introduce these two heterologous genes. This led to a only moderate improvement at the first trial over the previous strain. This is likely due to the fact that in Saccharomyces cerevisiae, glycolysis is the predominant pathway. So the glucose and via glucose 6-phosphate is degraded mainly to ethanol in yeast in Saccharomyces cerevisiae, and only a minor fraction of glucose 6-phosphate is going to the pentose phosphate pathway, which delivers xylulose 5-phosphate and NADPH. So our next goal was to enhance this pathway and decrease the glycolytic flux. And this was done by 3 modifications, which were done after each other. First, we downregulated the phosphoglucose isomerase to reduce the flux in this direction, and overexpressed the glucose 6-phosphate dehydrogenase encoded by the ZWF1 gene. And also we deleted the ALD6, the acetaldehyde dehydrogenase, which is the other source of NADPH in yeast, to force the cell to go through this pathway for producing NADPH. So in this way, we should get both a higher production of NADPH and of xylulose 5-phosphate compared to the starting strain. So when this was done, we get indeed a further improvement, which still was rather moderate compared to the previous strain, but it was a 3-fold improvement compared to the starting strain in which only the mutated fatty acid synthase was overexpressed. So this shows that indeed this traditional metabolic engineering can already increase the pathway productivity to a certain extent. But there was also an idea to target the secretion of octanoic acid as the next target beyond metabolic engineering, because this secretion on one hand facilitates the product purification, on the other hand it also may increase production by a thermodynamic pulling force, because octanoic acid is removed out of the equilibrium from the cell. It also may help in reducing the toxicity of the product which accumulating octanoic acid could induce. So an approach was done by the colleagues in Chalmers, which are also our partners in CHASSY. They created a randomly mutagenized library of the TPO1 gene. TPO1 is a transporter which is known to transport also medium chain fatty acids. And they subjected it to random mutagenesis by aeroprone PCR to isolate variants which have an improved ability to export fatty acids in a chain length range from C6 to C14. So they mutagenized it, cloned it into a plasmid, and transformed the library into a non-producer strain. And this strain was exposed to increasing amounts of medium chain fatty acids, so that strains which had an increased ability to tolerate these acids were enriched. And from these strains, from the best clones, plasmids were isolated and sequenced. And they found several point mutations in the transporter sequence, and in the best variant, called M49, 3 point mutations in the transporter sequence were found. So when this variant now was integrated into a producer strain, able to produce fatty acids in the range from C6 to C14, they found that the extrusion of these fatty acids was improved compared to the reference strain when the wild-type transporter was expressed. So especially for the longer chains, for C14 and C12, there was an improvement, but also for the shorter chains, for C8 and C6 also, at least when the transporter was expressed in 2 copies. So this demonstrates that also transporters are very promising targets in engineering chassis strains for production of medium chain fatty acids, and possibly also of other fatty acid-based products. So all engineering approaches which I mentioned so far were based on the knowledge, on logical targets, let's say, on metabolism and degradation of fatty acid, on transporters which were known to transport them. But of course, there are also many factors inside the cell which are not known, or whose role we do not understand in the context of a particular chassis strain. So to isolate such factors, we reasoned that high throughput approaches should be applied using a biosensor. And for this purpose, we developed a biosensor which is based on the PDR12 promoter. So this promoter is known to be responsive to medium chain fatty acids. It is activated by a transcription factor called VOR1, which responds to medium chain fatty acids and thereby activates the promoter. So we fuse that promoter to GFP as a readable output, and then we measured the response of the sensor to increasing concentrations of octanoic acid. So here in the left panel, a non-producer strain was exposed to increasing concentrations of octanoic acid, and by fluorescence activated cell sorting, we could show that the response of the biosensor occurs in a concentration dependent manner. So the biosensor approach was obviously feasible. We could also confirm this by integrating the biosensor into a producer strain. And when this is compared to a non-producer, we see that both populations can be separated very nicely using FACS approaches, so that also this system can be used to screen producer strains for factors that improve octanoic acid production. To screen different factors, we selected a plasmid-based library covering the whole genome of Saccharomyces cerevisiae. We transformed that library into our producer strain, which also expresses the sensor. And via FACS, we performed several rounds of enrichment and selected clones which exhibited the highest fluorescence in this way. So next, we have performed fermentations with those strains to validate the effects of the plasmids on octanoic acid production. And we could observe that indeed 2 of the plasmids contained genes which had a beneficial effect when overexpressed from a plasmid on octanoic acid production. So here we have the KCS1 gene, which is the inositol kinase, and in the other panel here we have FSH2, which is a putative serine hydrolase. And both of these genes have themselves a positive effect, but especially when they are combined. So there's a cumulative effect of these 2 genes. And since both of them were not implicated in fatty acid biosynthesis before, this nicely demonstrates the potential of using biosensors and high throughput screening for engineering chassis strain beyond the traditional metabolic engineering. So with this slide, I would like to end my presentation and take the people who were involved in the Octanoic acid project, Eckhard Bohlis, and the PhD students Leonie Baumann and Florian Wernich, who performed all of the work that I presented. And of course, to the whole CHASSY team led by John Morrissey and the other PIs here. Thank you very much also for listening, and I will be happy to take questions.
Thanks very much, Mislav. Very nice talk on your work on fatty acid synthesis. So I'm going to take a look at the questions now. And so if you have a question, you can type your question into the Q&A. I might actually start with one myself. And the work on engineering the fatty acid synthase to go from the C16 to the C8 is obviously really nice. Can you also go the other way? Can you go from C16 longer? And in an overall sense, how predictable and programmable is that system? Or is it a little bit empirical and you just have to try things out?
Well, it is to a certain extent certainly predictable, because all the mutations which we performed to decrease the chain length were predicted to have this effect based on the known 3D structure. To my knowledge, it was not attempted so far to engineer it for longer chains, but in nature there are fatty acid syntheses which are also able to synthesize longer chains, like for example the one from Mycobacterium vaccae, and for example by comparative studies but could also try to infer which modifications could maybe be translated to a fatty acid synthase from another organisms to achieve the synthesis of longer chains. So it's certainly, to make it short, it is certainly predictable to a certain extent how these enzymes work and how they can be engineered.
Okay. I actually have a series of questions I had over. They've all been coming in. So maybe on a slightly similar topic, it says how generic is your engineered chassis? And the question is how easily can it be adapted for producing designer fatty acids? So for example, branch chain fatty acids or other specialized fatty acids.
Yes, so regarding the precursor supply and cofactor supply, this should be generic for all fatty acids, because the fatty acid biosynthesis shares the same precursors. However, the degradation pathways partly vary. For example, the activation of fatty acids in peroxisomes by ligating them to coenzyme A can involve different enzymes depending on the chain length. So the engineering or preventing the degradation could be more tailored to the chain length, and the precursor supply pathways are rather generic.
Okay, and there's a question. I think one of these questions maybe. Did you identify? I mean, I guess again it's similar, but did you identify fatty acid synthases that can produce fatty acids in the C10 to C14 range specifically?
Yes, there are also such variants, also published by Chalmers partly, but also in our work, there are variants which are able to produce C10, C12, yes. Although not exclusively, they are in most cases producing mixed population of medium chain fatty acids. So most of them are not really exclusively producing C10.
Okay, and maybe just following up on that question, then there's a question asking about the purification, because I guess the person is noting that in the graphs you showed, the strains weren't producing only one fatty acid, there's mixtures. Does that create problems for purification, thinking about scale up and commercial production?
Well, the mutant which we used here in the work that I showed is quite specific for C8, so they only minor amount of C6 and C10. But certainly the purer the better for the downstream processing. We have not yet developed methods for purification, but the chain lengths of course tailored in this case. So we have mainly a production of C8, which I think should be also then relatively easy to purify from the other byproducts, which are only produced in minor amounts.
Oh, okay. So thanks, Mislav. I think I'm going to have to stop there. There's a couple more questions around biosensors, but hopefully we'll get to discuss the biosensor more maybe in the general Q&A. Okay. So thanks very much, Mislav. So you can stop sharing whenever you wish. And it's my pleasure now to introduce our third speaker today, Dr. Oksana Bilyk, who's a research scientist working in the University of Manchester, working on a TOPCAPI project. And Oksana carried out her PhD in Germany, working on Actinomycetes, and actually has a really nice track record of working on Actinomycetes and clusters of genes, biosynthetic genes, on antibiotics and on polyketides. She was a Marie Sklodowsky-Curie Action postdoctoral fellow in the University of Cambridge prior to coming to Manchester. And I'm really looking forward, Oksana, to hearing your presentation this afternoon. Thank you very much, and the floor is yours.
Thank you, Zhao, for the nice introduction. I'm going to share my screen now. Can you see it?
Yeah, that's looking good, and it's just going to present. Yeah, that's perfect.
Okay,
thank you.
Perfect. Okay, welcome everyone to my talk tonight. So I'm a postdoc, as John said, at Manchester Institute of Biotechnology, and today I'm going to present the work that we did as part of the TOPCAPI project. So TOPCAPI is an international collaboration between academia and industry. So there are 8 partners on this project, 3 universities, 2 research institutes, and 3 companies. We are based in 4 European countries that are Spain, Italy, Slovenia, and the United Kingdom. And the main aim of this project is to harness biosynthetic potential of actinobacteria, and in particular Streptomyces, in order to generate the production chassis that could be used efficiently for production of valuable compounds, and in particular antibiotics. So in this project, we basically use the design, build, test, learn cycle that is common in synthetics biology. But unlike traditionally, when it is start from the design stage, in TOPCAPI, we decided to start from the test stage. And we did this because prior to starting building the pathways and engineering our host, we wanted to learn as much as possible about the native host and about the heterologous host that we are going to use. So to do this, we start with the cultivation of our reference strains that are the native producer and the desired heterologous host. We do everything in industrial settings, so the strains are cultivated in bioreactors, and at certain time points, we collect samples to perform omics data. So we've collected huge amounts of data for both strains, and we perform targeted and untargeted metabolomics, transcriptomics, and also genomics. Once this data is obtained, we feed it into our pre-designed genome-scale metabolic models. By doing so, we can understand in which metabolic states are the cells during the production of the compounds that we are interested in, and also we can understand which pathways, metabolic pathways, can be and should be rewired within the cells in order to benefit production of the desired compound, but without affecting the accumulation of the biomass. Once this data is collected, we proceed to the build stage that involves pathway engineering and genome editing. So each partner on this project is an expert in certain area. We all work together to complement expertise of each other and also to move within this cycle. So here in Manchester, we have 2 groups. One group is led by Eriko Takano, and this is a group that works on synthetic biology, and the systems biology group is led by Rainer Breitling. So as John said, I'm a postdoc in Eriko's group, and therefore my work is mainly focused on the build part. The bacteria with which we work are, well, in my opinion, they're really beautiful. They are named Streptomycetes. They belong to the phylum of Actinobacteria. Here you can see the model strain, Streptomyces silicolor, producing this blue pigment, and so they're gram-positive bacteria that live in the soil. And I think all of you have encountered this bacteria or kind of experienced it if you've ever been to a forest after the rain. So there is this particular forest smell after the rain, and it is because of the presence of geosmin in the air. And this sesquiterpene is produced by Streptomycetes. So Streptomycetes have very high GC content. It's around 70%, which basically doesn't work them very easy bacteria to work with, but they're also very talented producers of natural products. So on average, 12% of their genomes is dedicated to the biosynthetic gene clusters. Moreover, over 50% of antibiotics that are currently on the market are either derived or inspired by Streptomycetes. However, only few species or only few strains of Streptomycetes are genetically amendable, and it quite often happens that if we start working with a new Streptomyces strain, we have to optimize the existing molecular tools to be used specifically in this organism. So in this project, as a heterologous host, we chose Streptomyces silicale because it is the best characterized actinomycete up to date, and so we can call it like a model Streptomyces strain. And the molecule that we are interested in is G2270A. So here you can see the chemical structure of this compound. And we are interested in this molecule because our industrial partners, Nikons, exploit this compound. So GE serves as starter compound for the semi-synthesis of an anti-acne drug that is currently in phase 2 clinical trials. GE originates from this rare actinomycete, Clonobispora rosea, which is not genetically tractable. So if you want to do any rational engineering of the pathway, it has to be moved to a heterologous strain. G2270A is a ribosomally synthesized and post-translationally modified antibiotic, meaning that the precursor for the final cyclized molecule is of peptide origin. So in case of GE, this pre-peptide is named PBTA. It is encoded within the biosynthetic gene cluster, and PBTA is elongated on the ribosomes as any other protein in its cell. So once it is assembled on the ribosomes, it can be used by the downstream enzymes in the pathway to give us the final molecule. So all genes required for the biosynthesis of GE are clustered within one locus on the chromosome, so they form this so-called biosynthetic gene cluster, which is very useful when we want to do heterologous expression. So when we started this project, the first thing that we did was integration of this pathway into the chromosome of Streptomyces silicale. So there was some preliminary data already available as other groups attempted to do this before, but the yields that we got from Streptomyces silicale were quite low, so it was less than 1 milligram per liter. One of the possible reasons for such low yields could be that silicale could be actually sensitive to this compound. So G2270 is an antibiotic. We decided to check how the strain behaves in the presence of GE. And indeed, there was no growth of the strain when we added antibiotic to the production media. So this was something that we didn't know when we started this project, and it was an obstacle that we had to overcome. So obviously, the next step was to generate a strain that would be resistant to the desired compound. So what does GE2270 do as an antibiotic? To put it simple, it inhibits protein biosynthesis via blocking the function of the elongation factor TU. So as for today, there is only one known resistant mechanism for GE, and it is modification of the target. So for example, in the native producer Pyruvate, GE can't bind to the EFTU, and therefore the strain is resistant. By knowing this, we decided to overexpress TUF, so the gene that encodes is named TUF, from the native producer in our heterologous host. However, as you can see on this bioassays, there was not much difference between this wild type and the engineered strain. So in this situation, we have mixture of both native TUF and heterologous one in the engineered strain. And of course, the wild type prevails. So another thing that we tried was to replace the native TUF from silicale with the one from Pyruvate. And to do this, we decided to use CRISPR-Cas. So CRISPR has been applied for Streptomyces before, but not to Streptomyces silicale. So we had to optimize this system. We've done this by tuning expression of Cas9. And after optimization of this tool, we could successfully swap the native TUF with the one from Pyrosia. So the obtained strain could tolerate high concentration of the antibiotic. However, the growth was impaired, which is not suitable for the chassis. So in parallel, we were performing the standard strain development. So we basically plated our host on the standard medium that was supplemented with the antibiotic. And after a few rounds of cultivation, we picked colonies that were resistant to GE. So this spontaneous mutant that we obtained grew very well in the presence of high concentrations of GE. The maximum concentration that we tried was 200 milligrams per liter, but there is no indication that it can't tolerate high concentrations of GE. So we decided to test this strain for the production, and when we incorporated the cluster into its chromosome, we could see at least twofold increase in the production. So at this point, we obtained the suitable host for GE production when we moved to analysis of the omics data. So we were cultivating the native strain under industrial conditions, and we obtained lots of data from it. So we performed this untargeted, targeted metabolomics, and also analysis of transcriptomics data. And here is just one example which I want to share with you today. So when we looked at the transcriptomics data of Pyrosia that was producing GE, we saw that PBTA, this is a gene that encodes the precursor for our final molecule, was highly expressed during the production. So we decided to overexpress it in the heterologous host. Another target that we chose was PBTR, and this is the cluster-satiated regulator. So both genes have been placed on the integrative plasmids and expressed individually in the strain, but we also tried an array of different promoters. All of them were constitutive, and the best result was obtained when both genes, PBTR and PBTA, were expressed together in our spontaneous mutant. So we had another look at the transcriptomic data from Pyrosia, and another interesting thing that we saw was that one of the ribosomal proteins was expressed at quite high ratio, and expression pattern of this ribosomal protein was different from the rest of the ribosomal proteins, but very similar to the one from PBTA. So we thought perhaps this ribosomal protein has some specific function during elongation of PBTA. To test this, we decided to overexpress this ribosomal protein together with PBTA in our host, in our heterologous host, and indeed there was almost twofold increase in the production. So we are not only doing gene expression, but we also attempted constitution refactoring of the biosynthetic gene cluster. So we played with the cluster quite a bit. We used both constitutive and inducible promoters. We inserted these promoters at different locations on the cluster, and at certain points, we had to completely change the architecture of the cluster, and here our partners at Explorer have been extremely helpful as the experts in the synthesis of DNA. So they synthesized for us this large DNA strands that we assembled into single constructs here in Manchester, and now we are testing them for the production of GE. So in summary, we successfully applied methods from both synthetic and systems biology to increase production of G2270A in Streptomyces silicola. And to do this, we learned a lot from the native host. So we collected different omics data, we analyzed it, incorporated it into our metabolic models, which helped us to choose the promising targets for overexpression in the heterologous host. So as for now, we have completed this, in our case, test, learn, design, build cycle, and we are ready to turn it for the second round to further increase the production in our host. So here I would like to say thank you to quite a few people. As you can see, there are a lot of us on the TOPCAPI project. This is a photo from our last face-to-face meeting. I also would like to say thank you to Takana Group, which you can see here, and of course SynBioChem Center that is located at Manchester Institute of Biotechnology, and who are sharing our facilities with us. So please follow us on Twitter, and you can always check our updates at our website, that is topcapi.eu. And thank you for your attention.
Oksana, thanks very much for sharing your work with us. That was very interesting. I'm just going to open up the Q&A. If you have questions for Oksana about that work, please type them into the Q&A. And you know, I completely share your enthusiasm for beautiful organisms that smell well. And I would say the only limitation with your system is that it can't make beer. But otherwise, you know, it's nearly as good as working with yeast. It was interesting, you know, the approach that you're taking. This is my own question now, so it'll probably be a bit disjointed. But you did a transcriptomics in, I'll call it the original organism that produces this particular molecule. And I guess you're identifying the conditions as well for maximum production. How does the level of production in the original organism compare to your heterologous host?
Well, at this stage, of course, at the at this stage in the native producer, we are getting much more. So this is industrial scale production where our collaborators can get quantities that are sufficient for purification and further use for semisynthesis. So we're talking about in terms of grams. But it is quite lower in our heterologous host right now.
And a kind of a naive question. I mean, would it not be easier to try and make the native producer amenable to genetic engineering, and you know, with all these new technologies, as opposed to completely moving the pathway? Or is that kind of a bit naive?
Oh, it's not a naive question at all. Actually, we do try to do this. So we didn't give up, and our collaborators didn't give up on this train right now. So we are trying to modify it to develop the tools. But the problem with Streptomyces is that they're not always fast growing. They have quite complex system, genetic system. So it takes some time to develop the tools and also to kind of persuade to work them with you. But yeah. So we are working towards that direction.
Okay, you're taking all the strategies. There's one question I see here. I think the person is commenting on the fact. I'm going to say it was easy. The person doesn't say that, or you didn't say that, but it seemed easy enough to get more resistant Streptomyces strains to this particular antibiotic. Is that a worry for use of the antibiotic in a clinical setting? Does resistance easily arise?
It wasn't that easy. So we had to cultivate the strain for quite a while to get the resistance. And we are also now sequence the genome. So we identify the mutations that could contribute. So we're trying to understand what is the resistant mechanism in this case. But it wasn't like easily spontaneous mutant. We put quite a lot of effort to get that.
Okay. Yeah, I kind of guessed that. Just on thinking about functions of different genes, I mean, do you have an explanation or a hypothesis for why overexpression of a particular ribosomal protein had the impact that you saw?
Actually, yes, we do, but we are preparing publication on this. So unfortunately, I can't go into more details because it is also, you know, on an industrial scale. So, but once the publication ready, I'm hoping we will be able to share these results with you. So we kind of have an explanation why particular this protein is important.
And if it's not divulging too much, might this be more broadly applicable to other?
I think this is an interesting question. I think that something that would be interesting to look into it. And I think it would be, if it would be broadly applicable, it would be applicable for this particular class of compounds.
Okay. And I see one more question I'm going to ask you, Oksana, before we move to the general discussion. And it's about your use of the multi-omics data. And there's kind of two parts to the question. The first part is to what extent did the multi-omics data inform the engineering strategy? I think you've partially answered that, but maybe you can say more. And then the other is there any other omics method that you would like to add to the assessment of the native host?
So omics data actually informed us a lot. So as I showed, for example, of expression of PBTA, was guided via transcriptomics data from the native producer. The same was true for the ribosomal protein. We are also working on targeted metabolomics, where we identified certain precursor and see if we could direct flow in that way.
Okay, that's great. So Oksana, thank you very much for the answers to those questions and for that very interesting presentation. So if I could ask the various speakers now to maybe turn on your cameras so that we can see that you're still with us and that you haven't snuck off to get coffee or anything else. You're all here. That's great. So we're going to open this up to a general discussion right now, and maybe you can type in questions that you have, and we can come back to some of the unanswered questions as well as we go through this session. But my first question is for Huimin, and maybe I might pass a similar question to the other speakers. And it's really how amenable are the approaches that you described to transfer to industry? Maybe especially when one thinks around something as infrastructurally with such requirements as the Biofoundry.
Actually, the Biofoundry is not that expensive, because you think about the cryo-EM, it costs more than $6 million, right? And the Biofoundry is just a couple $1 million, But the question that I don't think it will be for an individual lab, but it clearly can be, a facility for many researchers in a single institution. And currently, there are many actually Biofoundries around the world, including the one, of course, in University of Manchester, and also, the list is growing. And then if you look at the industry, right, there are also many companies, now moving toward that direction. Zymergen, Ginkgo Bioworks are the two leaders, in this field. So that's why I said, there are a lot of opportunities, because now we have the infrastructure to do many things that, cannot be done in the past.
Mm-hmm. Great, thank you. And Mislav, I might put a related question to you, and maybe the question is in terms of the kind of approaches that you're taking. What are the challenges maybe to transferring that to industry, transferring that technology and that approach?
The challenges are, of course, the yields, which are still achievable. The production is currently limited, likely by toxicity of the short chain or medium chain fatty acids. But yes, certainly I believe when we create chassis organisms with a higher robustness, that this could be feasible also in a larger scale production.
Oh, okay. So yields are obviously a big issue. And on that idea of transferring the technology to industry, Oksana, I think that if I'm right, that your partner company, Nakions, is using the native host at the moment to produce the molecule for the clinical trials and so forth. And how much better would you need your heterologous host to be before they would switch? Or is it a case that their strain is good enough at the moment, and it's really more a case of just trying to be better, just for a little bit more on the economic side?
I wish it would be, good at the heterologous level now, but in terms of a heterologous host, we are talking milligrams per liter, whereas in terms of the native host, this is gram per liters. So there is room for improvement, but so Streptomyces silicalis is a strain that we use. It is related to the native host, but not very closely related. And there is another potential host that has been published before. It is Nonomuria strain, and this strain has very large potential for production in heterologous, as a heterologous host for this compound. So this is it's another area that we are trying to cover right now. So everything that we learned in Streptomyces, we want to try to transfer to Nonomuria. Mm-hmm. So the host is just closely related to the original strain, and the general environment of the strain would be maybe more suitable for production of GE.
Oh, okay, thanks. And maybe I'll come back to Huimin with the next question. But in your experience, do you, you're very interested in both Saccharomyces, I'm thinking of yeast now, but of course I know you work on other platforms, but Saccharomyces and non-Saccharomyces strains, alternative hosts. And you mentioned that it is relatively easy now to, at least with certain yeast strains, to optimize other yeasts for production. But do you have any sense of how industry feels about this, and how willing industry is to switch from, the strain like Saccharomyces cerevisiae that's really well known and everybody feels very comfortable with, to using organisms that lots of us can't even pronounce?
Indeed, yeah. I couldn't pronounce all those species names as well. But I know that some companies also already use non-model organisms for production, wide variety of products. Just they couldn't, apply metabolic engineering or genetic engineering tools to further improve the production. Typically, they just use the traditional approach, like introduced kind of a random mutagenesis, right? Do the screening. It's just very slow process. But I think that now with the development of the CRISPR technology, they probably will apply those tools in their platform already. It's not like this is new to industry. I would say industry has been used those non-model organisms for many, many years. The one example, of course, just given by Oksana, right? And I think you can adapt the CRISPR tool for that organism as well, and then they can do the engineering in the native host.
So you would say, that probably there's not necessarily a reluctance of industry, and if we can make non-native, or if we can make non-traditional organisms produce higher amounts, in industry would be willing to use those strains.
Exactly. It's just that they don't have the genetic tools to do it. That's why you have to try heterologous expression. But I think this will change, with the new development of those new tools. Of course, the CRISPR also will not work in any organism, as we all know. There's also a lot of adaptation as well, right? And also, we actually also encountered a problem like rhodosporidium tauruloids. I mean, so far, there are quite a few groups work on this organism for many years already, but there's still no episomal plasmid available, right? So we actually spent a lot of effort to try to find those ARS and SAN sequences. And it is challenging. For some organisms, it's easy, you can identify them, but some actually not. Then that is still a limitation,
Yeah, of course, it does give the opportunity to do really interesting biology on those organisms, but I think industry would like solutions maybe faster. Eriko, do you want to come in and ask?
Yes. Hello, thank you very much all for the nice presentations. I was just wondering, it's related to what you were saying, Huimin, that you have the Biofoundry, you can use model organisms. On the other hand, there are companies who are using non-model organisms. Which do you think is better or faster or more efficient, let's say, in trying to get that product? Is it the old style, traditional, UV mutagenesis and then screening for new things? I know we're not there yet, but to try and diversify the chemical space, are we getting closer to compete with these traditional methods, do you think, Huimin?
Yeah, as I mentioned before, I think if the non-model organism, because the reason actually people choose non-model organism because they already have some unique physiology or production product profiles, right? They can, like for rhodosporidium or those Yarovia, right, lipolytica, they already can produce a lot of tags or the lipid-based chemicals. So then, if we have genetic tools, I think those organisms really represent a very good starting point. And if I compare it to just engineer the Saccharomyces cerevisiae, try to build the machinery and then overproduce the fatty alcohol-based chemicals, it's just more challenging. And but of course, if you don't have those genetic tools, then you have no choice. So I would say that it really depends on how soon we can get those genetic tools for the non-model organisms.
So you would still think this synthetic biology or metabolic engineering, metabolic engineering pathway will be the future, kind of?
Yeah, I actually, because I worked with the E. coli and the Saccharomyces cerevisiae, of course, for many, many years, right? And we developed many, many tools. But now actually I'm more lean towards those non-model organisms because indeed they have a very unique, the physiology, right? And also the capability to produce a targeted product at a very high level already. So if we just need to develop if we can develop those genetic tools within a year, right? And then I think there's no reason to work with the model yeast. Except for fundamental studies, of course, you still need a study. Yes. That's what we do right now. But if you really want to talk about the industrial applications, I see that.
Huimin, I'm editor-in-chief of Famous Yeast Research, so don't upset my my fan base by saying there's no need for yeast.
No, there's not. I didn't say no need. It's just for fundamental research. Definitely, is the model yeast still is the best, mainly because it has all the data. We also do like genome scale modeling for all the fundamental studies. Indeed, the model yeast still has a clear advantage because you have so much data, accumulated over the past few decades, right? I mean, no comparison. So much data. Yeah. It's just Rico just asking for industrial application, right? Yeah. That's different.
Yes.
Yeah. Yeah. And at the next webinar, we'll probably come back to this topic because we are going to be talking about validation and industry scale up, and there are of course challenges there as well. There's a few questions on biosensors, so maybe I think these are directed in the first sense towards you, Mislav, but then I think also maybe towards Huimin. And there's a question about the octanoic acid biosensor that you described. And the kind of the two questions I think are, one is whether you can make it very specific for octanoic acid and not be activated by other weak organic acids. And the second question then is asking about the concentration range at which it works, especially the higher concentrations, and how much of a, well, they don't ask, but I'll ask how much of a limitation is that?
Yes, I will first answer the last question, which is easier to answer. So the linear range is up to 100 milligram octanoic acid for the currently used biosensor, which I showed here. So it can be used only in a low producing strain, but of course the idea is to identify target genes in a low producing strain and to transfer them into a higher producer. And regarding the specificity for octanoic acid, this is something that we of course have also thought about, which is an interesting idea. Unfortunately, there is no crystal structure of the BOR1 transcription factor to my knowledge, so that no structure-based mutagenesis could be done to change the specificity, but we could certainly use approaches like Professor Zhao presented for some random automated approaches to identify also such variants of transcription factors, which could then also improve screening. Indeed, we also isolated clones which produce more hexanoic acid in our screens, which I did not show here because this was not the topic, but yeah, the biosensor can also be used for screening other short and medium chain fatty acids. Okay. And specificity will be an important target for the future engineering.
Yeah, great, thanks. And just staying on biosensors, and then maybe Huimin, but maybe anybody else can answer as well. But the question is, it's around using biosensors in biofoundries. And the kind of question, is there a consortium that develops these? But maybe more generally, I suppose, Huimin, could you comment on the use of biosensors in a kind of a biofoundry and automated strain development setting?
Yeah, the biosensor actually is pretty useful, and we have been trying to develop in vivo biosensors for several years. There are several types of biosensors. One is based on the transcription factor, just like Mislav mentioned, but also like a promoter-based, right? And also, or could it be like the ribo switch, those kind of aptamer-based one. Mm. But the problem is that it's just very difficult to develop those biosensors for the targeted compound, right? And I don't think there's like a consortium, to develop various biosensors. If there's such a one, that would be great. But I know there are quite a few groups are working, on biosensor development.
Yeah. We should make a consortium then. For the EU. A good idea.
Yeah. I mean,
it definitely will be very useful. Yeah, that's for sure. Because the one major limitation in the fully designed design, build, test, learn cycle is the detection, right? Currently we only use the Plato readers, yeah, it's very limited. If you have the biosensor, then really can take advantage of the genome scale engineering tools we have.
Yeah, and I think also maybe intracellular biosensors for dynamic regulation is something where there should be great potential.
Yeah, as well. Yeah, for gene regulations, exactly.
Yeah. There's a question I see. I'm not sure I fully understand it, but I'll ask it, and Huimin, you might understand it. It's this one about the algorithms, and I think that this is relating to the automated strain construction, or maybe to the genome scale models, and it's actually asking whether they take into account other aspects of the cell factory, such as byproducts or propagation or other things.
Yeah, the machine learning algorithm we actually used in pathway optimization is very simple. It's very actually kind of the standard one. It's a Bayesian optimization. Right? So we didn't take account any other factors. It's really just look at the data, right? How actually they can, what kind of combinations can lead into higher production, etc. So we're not taking any other factors into consideration.
Okay.
But I think that is the area that need further development. Exactly, is that all the omics data, right? How they can be incorporated into the machine learning model to predict those non-obvious gene targets.
Yeah, absolutely. Enrico, I think you want to come in.
Yeah, is that okay? I know time's going, so maybe perhaps one of the last questions, perhaps. But I wanted to ask to all three of you. This is the building part of this design, build, test, learn. And what do you think is, or rather, where are we going in the future? What is the technologies that's going to come in for this building area? We've got a lot of toolkits, like Huimin said, with lots of assemblies, robotics. We couldn't do these things before, even CRISPR doing genome editing. But what's the next step? That golden next step that you think will happen? All three of you maybe could say some comments on this future of the build side.
It's hard to predict the future.
Actually,
I only know the general direction, right? As I already mentioned, it's really the combination of synthetic biology with automation and machine learning. That's the kind of general direction. But if you really want to say that what kind of a new technology can appear in the build component, because no one actually predicted the development of the CRISPR technology, for example, right? And we have been working with the genome editing for a long time, but we mainly used the Huimin nucleus, right? And then suddenly the talent popped up, and then CRISPR popped up. So no one can predict those things. But I think in the build component, we do need a kind of a general applicable tools, but also standardized tool. Because even for CRISPR, you still need to do a lot of adaptation, and it's not that actually that easy. Actually, still not that easy. For non-model organism, that's easy, but for non-model organism, still a lot of work need to be done, right? And then even for the construction of large DNA molecules, that I think is still challenging. It's not like, of course, we have so many assembly tools, but can you really build any DNA molecule for any length? I think that's still a big challenge, right?
Yeah, perhaps Mislav and Oksana has also some comments on this, what their vision.
I agree with Huimin that although we have all the tools, it looks like we have all the tools, we still can't do many things. But I think it would be also very good if we could understand the regulation, how it happens on a better way. So because, well, if we could also incorporate it into such tools as heterologous expression, that would be very advantageous.
Yeah, thank you.
I also think that we are very far away from completely understanding even the simplest bacterium, and this is in the future to come to get a deeper understanding in the physiology of our strains, of our model organisms, which finally will also, in combination with the novel tools, make the further progress possible. So I'm not only on the tool side, but also believe that basic science will also contribute a lot of new information to strain engineering.
Yeah, thank
Yeah.
you.
But this is difficult to predict.
Yeah, you know, I mean, on that, I think it's become apparent that there was about, I'll say, a 30-year dark age of microbial physiology. And, you know, so often now when we're looking, especially at non-traditional organisms, the place where we find the information of physiology are papers in the 50s and 60s or even 70s, and then nothing, you know, because there was a complete lack of funding for that area for a long time. And it was only when systems biology, this is a personal opinion now, I think, but only when systems biology started coming and the people started trying to understand the whole cell again, we realized, gosh, there are no more microbial physiologists out there. You know, they were a dying breed. And I think that's coming back. But I think we lost a lot of years in understanding cells. And we have to get that back. And that's really fundamental research, as you say, Mislav. But maybe fundamental research now enabled by all these new tools and technologies. So it is exciting. And I think microbial physiology is to the forefront again. It's really important. I'll just say, so no one's left out, there was one question, but I'll just make it as a comment because we're short of time. And the comment is, of course, for bringing tools into non-traditional organisms. Transformation often is still a challenge, which is kind of ironic because, you know, we've been transforming organisms for a long time. And it's very frustrating sometimes when you switch to another organism. And I can even say with Pluvomyces that we work with, some strains just won't transform and we don't know why. And so that's a very frustrating thing. So I'm going to share my screen now because I would just like to kind of wrap up. I'd like to thank our speakers, our three speakers, for inspiring talks, really nice talks. I've no doubt that everybody who listened really enjoyed the talks today. They were tremendous. So I really appreciate the effort you went to prepare such nice talks as well, because as I said earlier, everyone's under time pressure, and we appreciate that. And I'll just do a little bit of promotion now, because as I said at the beginning, today was webinar 2 in a 3-webinar series. We had a design webinar. Today we had build, but we already today started touching on the topic of the next webinar, which is transferring all of these great strains that we can develop to an industrial setting. And that will be the topic of webinar 3 on the 8th of April at 1700 Central European summertime. So you can register at the same place you registered for today. If you enjoyed today, as I say, tell your friends. Please feel free to tweet or email about this webinar series, because we're very happy to have as many participants as possible. On the 8th of April, our keynote speaker is going to be Dr. Sophie Rolens from the Biobase Europe pilot plant in Belgium. And I'm really looking forward to Sophie's talk, because there are many molecules have been scaled up in the Biobase Europe plant. And while I don't know how much Sophie can talk about specific case studies, I think she'll certainly be able to give us a very good picture of what the challenges are around scaling up microbial cell factories towards industrial levels. And then we have 2 other speakers, one from CHASSY and one from TOPCAPI, who are going to talk about case studies of validation and scale up from within their own organizations. So with that, I will stop and say thank you very much, everyone, and have a very nice evening, and well done to all our speakers. Thank you. Bye-bye.
Bye-bye.